We're very happy to present to you today a series of talks from three of our faculty here at Garvan. Unfortunately, you'll have to hear from me, but as well as a couple of other speakers from Garvan in various aspects of the immune system, as well as an invited speaker. We have a very special guest, Cheryl Van Noonen from North Shore Hospital, who will tell us about her fascinating work in tick allergy. So the immune system, I think, is something we all need. The boy in the bubble, as Stu Tangi might mention later on, can't get around in the world because he doesn't have an immune system. So most of the time it serves us very well. It protects us from infections. One of the great emerging themes in immunology is the job that immunology does in protecting us from cancer a lot of the time. And one of the great developments is, I think you'll see over the next few years, is the development of ways of harnessing the immune system to fight cancer. That's one of the exciting developments and something we're working on here in conjunction with the cancer division here at Garvan. But the immune system can also cause us some problems. When it doesn't work, we can have immunodeficiencies like the boy in the bubble. When it becomes overactive and targets the wrong things, like parts of our body, which it shouldn't destroy, starts destroying, that's when we have autoimmune disease. And inappropriate reactions against things like airborne allergens and food allergens, of course, can also cause us problems, and we'll hear a bit about that today. So the immune system is our friend, but we have to learn to live with it, and sometimes it misbehaves. So some of those instances are a bit about what you'll hear about today. Okay, so without any further ado, I will kick off proceedings, and what I wanted to tell you about today stems from some of the work we've been doing in the lab my interest is in a set of cells called Bcells, which make the antibodies which protect us from infection a lot of the time, and really are what we want to get when we have a vaccination to protect us from reinfection with all sorts of things. But as I said, antibodies can also cause diseases, autoimmune diseases and allergy in particular. And so some of the work we've been doing in the lab has been looking at the very basic mechanisms which control the cells which make antibodies and how they can go wrong in autoimmune diseases and such as lupus, and in allergies such as asthma. And what we found was quite amazing to us anyway, a connection between these two types of diseases and the basic ways that these diseases can emerge. And what I'd like to get across to you is that this very basic research is really meshing with what we're seeing in the clinics now and new clinical trials, where we're seeing a connection between these two mechanisms, which has the potential to give us some new insights into treatments for these sorts of diseases. Okay. Okay, the antibody. So antibodies, you've probably all heard about. This is the famous Y-shaped structure of the antibody, which was defined in the 60s and a couple of guys won a Nobel Prize for. There's been quite a few Nobel Prizes around antibodies, their discovery, their use in various ways. And there's no doubt about it. It's one of the major tools in the biopharmaceutical industries, and of course very important for our protecting us from infection. So just a few basic things about antibodies. This is a protein, so it's a very small thing which will circulate around the body, and it has two major components. It has up one end these paired regions, which we call variable regions, and they're called variable regions because if you look at one antibody and look at another antibody, these variable regions will be different. And the reason they're different is so all your antibodies are basically different. That's because some will want to bind to and eliminate a virus, for instance, or a bacteria, or anything else which is wanting to be eliminated from the body, or if you're unlucky, it may react to a part of yourself, so it may be an autoantibody, or it may react to an allergen, such as found in peanuts and food allergies, in which case it may be a pro-allergenic antibody. But that's all about how the antibodies stick to something, and I'll show you just a bit more about that in a second. And the other component, which I'll talk a bit towards the end of the talk, is what we call the constant region. Clearly it's the same between antibodies, but not all the time. There are basically different classes of antibodies which share constant regions and so share different functions. And so we'll get into that in a bit more detail. So this guy is a stylized B cell. As I mentioned, that's the cell of the immune system we talk about. So these are circulating around your blood, but most of them are found in your lymphoid organs, such as your lymph nodes, which you feel swell up here and here, or something when you have an infection, in your tonsils, if you've still got them, and then your spleen is another huge source of them. But what the basically specialised job that these guys have is to, once an infection comes into the body, they start producing these antibodies which can stick to the invading virus, bacteria, and the process of them sticking to it removes it from the body, neutralises it, and gives you long-term immunity should it come back again. So here's a photo we took in the lab of inside a blood vessel, and what it's meant to show these guys here are beautiful red blood cells, which is the majority of things in the blood, and but what you also see in the blood are these white guys, these white blood cells, some of which are B cells, but are also a whole bunch of other immune cells. And what you can also see, of course, here are the antibodies. And so antibodies are found in all our bodily secretions. There's a lot of it in our blood serum, in the component of blood, which isn't the cells, but also in all our bodily secretions really have antibodies in some levels to protect us against infections, which typically come in through the surfaces where secretions are. So if you've had a vaccine, what you'll do is have a lot of these antibodies raised against the vaccine, which are floating around. And so if you've had a tetanus vaccine, the tetanus bug comes into you, they're there ready to get rid of it. And so here we go. So here's another picture we took of a virus cannonballing down the blood vessel. And what happens is that some of these antibodies, not all of them, but a fraction of them, will have these variable regions which will stick to components of the virus. And sort of changing pictures, we had to take a picture of someone else for this one. But and this is a slightly different virus, as you can see. But so there are structures on the surface of the virus or the bacteria or whatever really, where there's a basic lock and key. And so particular antibodies which have the variable regions which are particularly suited for the purpose will latch on to these guys, and this starts a whole process which there are many ways it can happen, many mechanisms which can serve to get rid of the virus or bacteria, but this is basically the initial step in these guys getting flushed from the system. So this is the major job which antibodies do. Okay, yeah, there's the virus. Okay, so that's what we want antibodies to do, but sometimes, as I mentioned, they're not always doing what we want, and they can lead to disease. And so one of the most frequent diseases which are due to the body's immune system attacking the body itself, the so-called autoimmune diseases, is lupus. It's also called SLE, or systemic lupus erythematosus. And it's a so-called systemic autoimmune disease. That means you can have organ-specific autoimmune diseases. So Shane Gray in the next talk is going to talk about type 1 diabetes, which is a very specific autoimmune reaction against the beta cells in the pancreas, your insulin-producing cells. SLE is a more systemic and affect lots of organs. And so it's this very characteristic rash across the face, this butterfly rash in lupus patients. It's actually no accident it's a woman. It's very prevalent in women as opposed to men. But there are a whole sorts of other complications, in particularly in the kidney in bad cases of lupus. And this is actually due to autoantibodies, antibodies which are reacting to components of the body itself. So similarly, this is an actually autoimmune disease. I mean, the inflammation of the skin is very similar, but this is a different class of disease. In this case, it's allergic eczema. And so this is a completely different mechanism, it's thought. So it's not an autoimmune disease, but it's due to an allergic type reaction instead. So what's common between these two classes of disease is the antibody. But what is different, and so it always involves antibodies sticking to something it's not supposed to via these variable regions. But what appears to be different between these two types of immune-mediated disease is actually the class of antibody. And so here, I've just done a great colouring job here, fill in the numbers there, and just to describe, these are these constant regions we were talking about before. So, within each class, the constant region is the same, but the B cells are able to actually keep the same variable regions, but change the constant regions as they respond in a disease. It's quite amazing the way in which it can do it. And so whilst the response might start off making IgG antibodies with this kind of structure hanging off the end of the variable regions, it can progress and what's called switch to make antibodies which have this different constant region which is called an IgE constant region. And so it turns out that you're going to have identical antibodies in terms of the variable regions, but with different constant regions. And they, what they do is enlist very different, what we call effector mechanisms in the immune response. The result of them binding to something recruits in different cells of the immune system to try and destroy the target that it's bound to. So to try and explain that a bit better, so this one's a little bit more complicated. So in the case of autoimmune diseases, such as lupus, these are strongly involved autoantibodies of the IgG class. You can see here, there are all sorts of ways that the binding of IgG to parts of the body. In here, we've got endothelial cells on the inner side of a blood vessel. And we've got IgG autoantibodies lodging at the membrane here, where these endothelial cells are lining. And what they do, once they lodge here, they form these, what we call immune complexes, and they're this target to recruit in other members of the immune system. Actually, most of the cells, white blood cells that float around in your blood, are actually neutrophils, these guys here. And so they're ready and primed to recognize this concentration of autoantibodies at the base of the membrane. And then what they do is release all these inflammatory mediators and start causing damage to the tissue here, because they're basically thinking if this is something that needs to be attacked, it's something foreign that needs to be eliminated. But because the antibodies are binding to part of self, and that's where something's gone wrong, the sort of targeting of the system has gone wrong, then there starts to be damage to the body itself, in this case, the blood vessels. And this is very typical, particularly in the kidney, of lupus. So in the case of allergies, however, as I mentioned, these are due in a lot of cases to these IgE antibodies. And the way they work is well, it's not completely different, but it's quite different in that there are particular cells in the body. One of them is a mast cell, the other one's called a basophil. The mast cells have a lot of them in the skin in particular. And what happens is they have particular things, what we call receptors on their cell surface, which don't actually recognize IgG antibodies, but what they do is recognize IgE molecules. And what happens when we have an allergy is that we've made IgE antibodies against an allergen, house dust mite, maybe something in your cat's saliva, and these antibodies stick, and typically there's only very low amounts of IgE in your serum, but it's very potent because this is a very, very, very strong reaction that the IgE antibodies can stick to the surface of these mast cells, and when they then bind to the allergen that's around in the food or the animal secretion, we get this release of histamines and other inflammatory molecules from the mast cells, and that's what leads to the inflammation, like we saw on that woman's face with eczema. So although IgG and IgE act in different ways, I guess in the end they're both sort of precipitating this inflammatory reaction, which is saying there's danger here, even if there really isn't. I mean, in both of these cases. Okay, so well, that's all well and good. So what does it actually mean in terms of disease? And so we in the lab have been working on and trying to figure out why the body makes antibodies that will bind to our cells, autoantibodies, and we know. And to do that, it's extremely hard to do in human systems. Not many people will be prepared to go through the sort of things required to sort of dig down and figure out these very basic mechanisms. So we used mouse models, which have a immune system very closely linked to humans. And what we've done is look at a mouse model where there's a particular mutation, an inherited mutation, in this molecule called FAS. And so humans with FAS mutations have very strong autoimmune type of diseases like lupus, and there's another one called ALS. And so we've looked in mice which have this mutation, trying to find out why these mice and why humans which have this mutation make autoantibodies like these IgG autoantibodies. And so you can see here, I'll tell you what the analysis process here is. A very, very sensitive procedure, but hopefully you can see here there's not very many of these IgG cells under normal circumstances, but in the FAS mutation circumstance, these are massively expanded. That's about 500-fold more. What we weren't expecting was at the same time to see a whole bunch of these IgE antibodies being produced in this model of the autoimmune disease. So these mice don't actually get an allergic disease, but they're making this stack of IgE. So we're thinking, well, this is kind of what we expected, but what's all this about? And, is this actually happening in humans? So we actually did have a look in humans with FAS mutations, and it's true, they have a lot of IgE, and no one's really looked at it because they had autoimmune disease. And so everyone's expecting IgG, but so no one's really looked for the IgE. And it turns out in humans with this mutation. There's a lot of it there, and so this has kind of triggered a lot of work in this area. And in fact, it's turned out when people look, as the group at the National Institutes of Health, led by Juan Rivera, he's looked at the SLE is the disease, and they found a whole stack of not only IgE in patients with SLE, but found that it's actually autoreactive IgE, which is something which is not involved at all in allergy, but seems to be playing a role in lupus, because the patients with more of this stuff have a worse outcome, etc. And so it seems that this, in conjunction with the work we've done, is really pointing to IgE as potentially being an important role, not in allergic disease, but in promoting this autoimmune disease, lupus. And so this clinical data paired with our basic mechanistic data, which potentially can give us a way of trying to control this, is really pointing, I think, to an interesting new idea of how we might treat this autoimmune disease and perhaps others. And I was really glad to see just recently that there's a drug, well, it's actually an antibody directed against IgE. A lot of new drugs are actually antibodies raised against particular things, but this antibody can actually neutralize IgE. And so based on these new data, there's actually a clinical trial started at the National Institutes of Health, trying this anti-IgE antibody to treat lupus. And so that's actually recruiting patients now. So that hopefully will have some good results and point to a new way of potentially treating this disease, which really has no good treatment at the moment, except for anti-inflammatory drugs, which are okay, but have a lot of side effects. Anyway, so I hope that just gives you a bit of an idea of how we can marry this sort of basic research that is done in some of the labs involved in medical research, all the way through to the clinical research. And the great thing is when you can push those two things together, get the clinical data from patients, and be able to look at it in models where you can manipulate things, get more ideas, and then bounce back into the clinic, and hopefully with that to and fro between basic and clinical research, get some real ideas in advancing treatments in disease. Okay, so that's it from me. What I'll do now is introduce to you our next speaker, who is Associate Professor Shane Gray. So he works here at Garvan, has for I think 10 years now, probably, yeah. And Shane's expertise has been in two areas, both of which are covered in his topic there. He's worked a lot in the autoimmune disease, type 1 diabetes, which is primarily probably not mediated by antibodies, but mediated by T cells, as Shane will tell you. But he also has worked extensively in the area of transplantation, and particularly islet transplantation, to try and help patients with type 1 diabetes. And transplantation has its own problems, because unless you have a perfect match, when your transplant goes over, you're going to get it rejected unless you can find ways of trying to sneak that transplant in and circumvent the immune response to that. Anyway, so please welcome Shane Gray, who's going to tell us all about his recent work on that.
Good morning, everybody. Are we all good? All awake? I'm trying to think, there must be like some sort of warning about, going on after your boss, like when they say in showbiz, don't go after children or animals, I don't know. But anyway, I'm doing it, so we'll see how that works. Yeah, look, I do want to say thank you very much for coming. And I think also as a scientist, as a medical researcher who lives in this beautiful, edifice to medical research, it's really a pleasure to have the opportunity to directly tell non-scientists about what we do. So it is really, I really thank you for coming and giving me the opportunity to talk about our work here at Garvan. So we're going to talk about, I'm going to give you a little story about type 1 diabetes and what that really is. It's a disease that children get, but then once you have it for your whole life. And to tell you about type 1 diabetes, I need to tell you a little bit about how sugar in your body is controlled, and I need to tell you a little bit about your immune system. And then I'm going to bring those two together and tell you a little bit about the disease, what we're doing here at Garvan to help prevent it and stop it, and some of the exciting sort of developments both in the basic research and in what's happening in the clinic. And again, from the perspective of what the Garvan's doing. Okay, so buttons. So sugar, how is sugar regulated? So you have to sort of zoom in to your body, open up your abdomen, and you'll find this sort of squishy thing called the pancreas. And it sort of runs along in here, along your intestines. And there's like another cartoon of your intestine there, and there's this sort of pancreas here. I'm sort of cut it long ways. And you can see one of the most clear features is this huge sort of tubey thing. It's a pancreatic duct. And one of the things the pancreas does, it's full of all these tissues that release digestive enzymes channel along this duct into your intestines to digest the food that you've had for brekkie, right? But there's another sort of trick to the pancreas, another secret that wasn't actually discovered till the 1850s. And this is a tissue biopsy from a real human pancreas. And what's happened here is we've added a chemical that will stain most of the pancreas blue. You can see that's not naturally a blue color, but you can see that pancreatic duct here. And all these little blue blobs here are the enzyme, or the clusters of the cells that secrete the enzymes that digest your food. But you'll see there's something else kind of different about the pancreas. There's a sort of blobby brown thing. And this was noticed by a German medical student in the 1850s named Paul Langerhans. So we kind of named it after him. So he's got that honor for spotting it first. And I guess it's like a spotter's fee, right? That's how it works in science. So we call these the islets of Langerhans, or islands in English. And what we've done here is added another dye. And this dye picks up the hormone insulin, and you probably know about insulin. And you can see that this structure is really particular because it has lots of the brown stuff, and the brown stuff's nowhere else. So these islets of Langerhans are really quite cool. They're a beautiful sensor in your pancreas that can actually measure second by second how much sugar is in your blood. And when the sugar levels go high because you've just eaten and food is now coming in the blood from your tummy, the islet cells will release this hormone insulin into the blood, and it travels with the sugar to the liver, and it tells the liver, hey, there's a lot of sugar coming through, we've got to get the body to start storing it to bring sugar levels down. And this is really important because sugar is literally the fuel for your body in the same way that petrol is the fuel for your car. I guess if your car runs out of fuel, you sort of pull up at the side of the road and call the NRMA and everything's kind of sorted. But if you run out of fuel in your body, that is not sustainable, that's lethal. And so it's really important that sugar levels are kept within very defined ranges. And that's what these pancreatic islets do. So, okay, so now we're going to have a little journey in the immune system. I'm going to steal a couple of those high-resolution photographs that Rob had, that his lab is expert in. I'm going to play them on you now. And so we know that the body has got this blood vessel system, and that's the highway of your body and the pathways by which the immune system uses to get around your body. And look, I'm a sci-fi fan, I have to admit that. I don't know if any of you remember this movie, Fantastic Voyage. I just love the idea, the spaceship thing, and they all got shrunk and injected into the blood. Remember, and they were pulled out of the eyeball at the end? It was really cool. So I always sort of imagine if you could get into a spaceship like that, or a little, some sort of weird submarine and be shrunk, what would it look out the front window when you're running through the bloodstream, right? And again, am I really loud?
No, it's feeding back, that's it.
I had a really big coffee this morning. Is that better? You can turn down your whatever. How's that? Is that better? You like that better? So if you look out the front, look, you've seen this. Rob was telling you this is what the bloodstream would look like in an illustration, but what's kind of cool about this, it tells you a lot about immunologists, right? So the red stuff, what do we call that? Red blood cells, right? The white stuff, there you go. And you know how we describe these? Go and have a guess. Donut shaped. You get it, you can see where immunologists are going. We're obviously a pretty crude lot, right? But the immune system is really important, as Rob said, right? And I think he made a comment at the start that, we need the immune system, we have to have it. But then he said sometimes it misbehaves. And, medical research is about how does it work normally, that's really important, but also what's going on when it misbehaves. And I thought, we're going to talk about how it misbehaves, but it's also important to show you some of the cool things it does. And like the immune system is anti-cancer, and this photo's a little bit low res for the big screen, but these white blobs, these are now the white blood cells, they're the immune system cells. And this here, plated out, sort of stuck to this surface, is a cancer cell. And these immune cells are attaching to it. And see how this one is kind of elongated? They do that when they're angry and activated. And these are actually dropping digestive enzymes and nasty free radicals and other chemicals onto the cancer cell to kill it. So that's one of the key functions of the immune system, is an anti-cancer defense system. The other thing that the immune system does is it eats bacteria that enter your body. This is cool. This is a real cell. It's called a macrophage. Sounds fancy, but again, going back to simplicity of the immune guys, macro means big, because it's a big cell, and phage means eat. It's a big eater. So again, you can sort of see how immunology sort of progresses. But this is in green here, this is a bacteria in all seriousness, and you can see that this guy's actually reached out this kind of weird pseudopod, and it's actually going to grab that bacteria, pull it inside itself and dissolve it. Right? It's pretty cool, right? So the immune system has all these incredible functions. This is real live image of an immune cell in green chasing a bacteria in red. It's from a colleague of mine, Tatiana Chatnova. But this is like really in real time how immune cells look. This is like a giant Pac-Man. Check it out. Chasing a real bacteria. Look, the bacteria is trying to get away. The immune system is chasing it. Oof, gone. And it's really amazing, right? I mean, immunology is fantastic. So that's when it's good, right? What about when it's misbehaving, as Rob said? And that's a really nice, elegant way to put it. So this is another sample, another biopsy from a patient. This time it's a person who has type 1 diabetes. And again, this is from the pancreas. There's going to be a test at the end of this morning session, isn't that right, Rob? So I'm assuming your pathology is up to scratch. So you can see immediately this is a pancreas biopsy. You can probably recognize the ducts here. But these are the cells that secrete the digestive enzymes. But there's your pancreatic islet, right? You can sort of pick that out. Looks pretty normal. This time we've used a different dye to highlight the tissues. It's pink, right? But what you see here is another pancreatic islet, but it looks really different, doesn't it? All those small dots, each one of them is actually a white blood cell. And we know they shouldn't be there. They've actually come there to destroy the cells that make insulin. If you put the brown stain on again to look for insulin, which we did in this biopsy, you don't see any brown, it's gone. So the cells that make insulin have been absolutely destroyed by the white blood cells. And here's another picture down here, we've added a stain. The brown here is not picking up insulin, it's actually picking up very specific types of white blood cells, or in this case the T lymphocytes or the T cells. And you can see these T cells here surrounding this pancreatic islet. So what is type 1 diabetes then? This is the sort of the fast facts, the take-home message. And this is again another image, another really beautiful colour image. This time we've got a fluorescent dye that's red that picks up insulin, so you can see your nice round pancreatic islets. And the green here are immune cells, these are T cells, and you can see they're all swarming around wanting to attack. So what is type 1 diabetes? Well, it's not the same as type 2 diabetes, they're very different. Type 1 diabetes is about an autoimmune attack. Your immune system attacks yourself and destroys those cells that make insulin. Once they're destroyed, meaning absolutely killed and gone, no other cell in your body or other tissue can make insulin. And so you can't control your blood sugar anymore. And your blood sugar can go crazily high and crazily low. And if it wasn't for the fact that we have insulin, it would be lethal. Right? That's the brute fact about it. But and it affects about 122,000 Australians currently. So why do we get it, and what's it all about? And that's what we're here one of the things we're trying to work out here at Garvan. Well, we know now that it's a genetic disease, right? It runs in families. You can also get it de novo, but there's about 20 genes involved. It's a kind of a curious disease because something's going on with the environment as well. It's not just genetic. There's an environment and genetic interaction, and that seems to be the case for a lot of sort of diseases that we get, these very complex interactions with us and the environment. But why do you get this? Why does it actually start? In a sense, what is the trigger? Like if you have the genetics and maybe the environment, is there something that precipitates it? And we still really don't know that, and that's probably the biggest question in the field. Because obviously if you knew that, you could try to prevent that from happening, or people being exposed to whatever that trigger was. So one thing we've been trying to do in Australia, what we have done in Australia, is to work out ways to prevent it but also treat it. And look, you do know if you've got colleagues or friends or family with type 1 diabetes, you know they take insulin, right? They take it with a needle and they measure their blood sugar. And that's fantastic. But there's a small percentage of people who for that is not really good enough. And they develop what we call diabetic complications. They get damage to their kidneys, they get damage to their eyes. They can also have a thing called hypoglycemic unawareness. It's a very technical term, but it means they can't tell when their sugars go too low. And they can fall unconscious. They can't work, they can't drive. It's a very disabling disease or very severe complication of type 1 diabetes. So for these people, current insulin therapy is not working. And so for this reason, we were thinking, well, again, fairly simply, if the immune system has destroyed your cells that make insulin, why not just put new ones back? And we know that works for, you can lose a heart and a heart can be put back and it works in a transplant model. So what we thought is, why don't we do islet cell transplantation? Take islet cells from organ donors and put them back into patients. And so the federal government set up this thing called the Australian Islet Transplant Consortium, which is actually now a national funded centre. And it had 3 sites, one at Westmead, which is kind of the lead site of which the Garvan was a part of, one in Melbourne and one in Adelaide. And I thought I'd just show you, so it's a pretty gory business transplantation, of course, but this is a bag which actually contains real live human islet cells that have been taken from an organ donor who donated their pancreas. And the islet cells are prepared into a solution, and then a small incision is made in the abdomen, a central line is put in, and the islets are actually infused into the portal vein, and there's all this sort of cool technology to track that the islets are going into the liver in the right spot. But this is what it looks like for a person who has type 1 diabetes and then gets an islet transplant. So the two key things, this is actually a clinical chart. Here is your blood glucose levels of this person, and the other really important thing really is this is how much insulin they were taking each day. So basically you can see their sugar levels go from very low to very high, really not very well controlled. And this is a small group of people with type 1 for which this treatment is designed. They get an islet transplant, TX1, bang, and you can see immediately already the sugars are really looking much better. They're coming down and they don't have so many big highs and lows like you're seeing here. And this is what we're going to talk about. For islet transplantation, it seems like you need a second transplant for it to work. Now, it's like saying going to get a kidney transplant and then a year later you've got to go get a second one to help the first one work. Right, so the technology is really amazing, but it's not working as well as we want. And this is one of the big research questions that we're interested in. But what's really cool is when this person gets the second transplant, you can see everything sort of looks a little bit better again. But look down here, this is the really cool story, right? The person goes off taking insulin, and this is like 4 years ago. So this is like one of the success stories of this transplant program. So what's the problem? The problem is, if you look at, there's your pancreatic islet transplanted now in a mouse in an experimental model. And these are immune greens, these green cells are the immune cells. And you can see as soon as we do the transplant, these immune cells are all over the transplant. They're sort of crawling all over it, like sort of bugs, right? You know, and the issue is that we need to be able to deal with that. You know, what are the interactions there? And if we could manipulate that in different ways, could we reduce the need to have a second transplant, and could we improve the success of organ transplantation in general, but also islet transplantation? So I'm going to take a chance here, assuming that you're quite sophisticated because you come to the Garvan sort of things a lot, and actually show you some real scientific data. So this is just one snapshot, and I'll try to keep it simple, but wave or yell at me or something if it doesn't make sense. But what I really want to show is how we're trying to manipulate the immune system and the sort of changes that we can get in the experimental models that I think in the future are going to lead to really big breakthroughs in the clinic. So in this case, this is what we call a Kaplan-Meier survival curve. And basically all we're looking here is we give transplants to mice and we ask, how long do the transplants survive? So if you give a transplant to a mouse, they all get rejected. That's normal. You have to give lots of immunosuppression for a transplant to survive. But what we've been doing here is we've been looking at the interactions. Remember the crawly bugs all over the islet? We've been adding anti-inflammatory compounds to the graft to see if we can change that environment. And when we do that, we get this sort of dotted line. So you can see the grafts do much better, right? We actually have about half the animals survive, and we don't give any immunosuppression. But in the clinic, immunosuppression is really important. And we thought that, one of the problems with immunosuppression, though, is it has a lot of side effects. But if you could reduce the immunosuppression, you might have the benefit without the side effects. So what we did is we went down to our local health food shop and dosed out immunosuppression into what we call a homeopathic level. So that's the green line here. So we add immunosuppression to the recipient mice, and you can see it doesn't have any effect on the transplant. When you put this new anti-inflammatory drug with the low-level immunosuppression, you get even better outcomes. And I think this is the sort of thing, this is the sort of breakthrough research that's happening at Garvan, where you can imagine what the future could look like and how that would meld in with current best practice to actually improve it even further. Look, these are the guys who actually do the work in the lab. And look, I really thank you once again for your time, and it's been a privilege to tell you about our research.
Thank you very much, Shane. Shane Stope's doing some fantastic work in this area, so I'm sure you'll agree. So our last speaker before our break is Carol O'Carroll from the Garvan Foundation, who's going to tell us a few words about the work of the foundation.
Good morning, everybody, and welcome to the Garvan. It's my great privilege to be working here at the Garvan as part of the foundation. I'll just tell you a little bit about what we do here at the foundation and what's coming up next year as well. And I'd just like to welcome you all again here today. And there are many of our regular supporters here. I recognise many faces, and also some of our partners for the future, who are people who are a very special group to us, who have left a gift in their wills to help us with the future work that's coming through the Garvan. So we're actually the fundraising and marketing arm of the Garvan Institute. And you may be interested to know that for every dollar that we receive from the government through the NH&MRC, the foundation has to find another 70 cents. And what we do with that is we fund novel projects and equipment. In fact, the work that you've just seen presented by Associate Professor Shane Gray was started up by philanthropic funds, and it became so viable that after a couple of years of runs on the board and raw data, they were able to then get funding through the NH&MRC to continue their work into clinical trials. So that was a very exciting development. In addition to fundraising, we also support the Institute with the public engagement program. So we put on these seminars each year. We also have a number of other functions, but we like to educate the community about the work that's been undertaken here at the Institute. And I think you'll agree it's a wonderful service that's being provided to the community to learn more about what's happening in the world of science here at the Garvan. For those of you who are new here today, I just thought I'd give you a quick rundown as to what the Garvan actually covers in the research areas. We've got 5 main divisions here. We do a huge number of projects in the area of cancer. Of course, we've got osteoporosis and bone biology, diabetes and metabolism, which is type 2 diabetes, and lifestyle and health and fitness activities that help with metabolism. Neuroscience, such things as Alzheimer's, Parkinson's disease, they're being studied here at the Garvan, and of course this immunology program that you're hearing about today. So as far as education goes, a little bit of more information. There's over 650 staff here at the Garvan, and over 600 of those are actually scientific people. 60% of our staff are female. You might be interested to learn. Yes, there you go, girls. Anyway, you've probably heard all that before. The average age of our researcher is 35. So I've done my best to pump up the average age here. And we've got them representing 53 different countries from around the world. So we're able to attract the brightest and best from all over the world to come here and work at the Garvan. So I know that many of you are already donating to us, but there are other things happening in the community to help our work, such as we've got corporate partners. The NAB have actually sent along some of their volunteers to help here today. And we do a lot of community fundraising. So we've got people who are shaving their heads, they're running, they're doing all sorts of crazy things. In fact, you've probably heard of Samuel Johnson, who's just ridden around Australia on a unicycle. Amazing, amazing feat that was, and very uncomfortable, I would say. But he was able to raise nearly $2 million in for breast cancer research. And of course, we've got people in the workplace who are still, actively working and providing funds through a donation that comes out of their regular salary pre-tax dollars. And that all helps a great deal. Other ways, of course, we've got our donors who are giving on a regular basis. We have appeal letters going out. We're always very careful with our appeal letters. We always ask people the first time they donate, how often would you like to hear from us? Because we don't like to inundate you with a lot of correspondence and aggravate you. So we're very careful about how often we talk to our donors. And a number of people are giving a regular gift just automatically as a deduction. And of course, now you're all ambassadors for the Garvan, so I expect you to go out and spread the word after today. Tell people how fantastic the Garvan is and what incredible work has been undertaken here. And of course, lastly, a gift in your will. And this is a very special way that people are choosing to support our work. And in fact, this whole area of immunology was started up by Bequest in 1998. And of course, I don't think we can all come to the levels of, funding a whole research area starting up, but any kind of gift in your will, a percentage of it makes a huge difference, and it all goes together in our endowment fund to help fund research well into the future for our children and our children's children. So when the time's right, I hope you'll consider thinking about leaving a gift in your will after you've looked after your family. So if you'd like to become one of our donors or supporters and receive our breakthrough newsletter, you've got the opportunity to fill in the form there on your seat and let us know that. You can receive it by email, which is always very quick and easy. We've got tours running here at the Garvan, and next year we'll have them running on Tuesdays and Thursdays. There'll be 2 of them a month. So hope to see some of you back again for our behind-the-scenes tours. Public seminars, of course, they'll be running again next year. There'll be 3 next year in 2015, but you'll need to check in December on our website to see when they are. But if you're one of our donors, you'll be getting that advance notice in your newsletter, and you can actually check in a book for all of them next year. And we have speakers who are available to come out and talk to your group. So if you're in a community group, we'd love to send somebody along to talk to you about the work. You may not know this, but we've got a fantastic website, and there's the website details there. And these seminars are actually all recorded, and they are up on our website in the area called Seminar Video Archive. So if you haven't caught up with everything that was said today, and you're thinking that you might like to catch up again and check the detail, you can go on our website and see it all later. We've got disease fact sheets available there, and they, as I said, you can find out about the seminars for next year. So could I encourage you to fill out that feedback form and give us some feedback about today, and also let us know if you'd like to be engaged in any of our other activities that are happening here at the Garvan. And yep, come up and say hello over coffee. I'd love to meet you all. Okay, thank you very much. Bye-bye.
Thank you very much, Carol. I can't speak highly enough of our foundation. They're a fantastic, committed bunch of people. So, please have a chat to Carol at the break if you want to. It is break time now, so if you'd like to either move out the back doors, front 5 rows, just out through these rows, please, and up the stairs around, and there'll be morning tea just upstairs there, and we'll see you in 20 minutes for some more immunology. Okay, welcome back, everybody, to the second half of our public seminar today. So we have two more great speakers for you in this session. And firstly, to introduce the first of them, who's our special guest from Royal North Shore Hospital, Professor Cheryl Van Noonen. So Cheryl is a senior staff specialist in the Department of Clinical Immunology and Allergy at Royal North Shore Hospital, and clinical associate professor at the Sydney Medical School at University of Sydney. So Cheryl's published quite extensively in the areas of allergy. In particular, stinging insect hypersensitivity, anaphylaxis, food allergy, and the basic signs of allergy, latex allergy. So I touched on IgE, and it's something involved with allergy, but Cheryl is a real expert in this area. And in particular, she's done some fantastic work in recent years on a really interesting aspect of allergy associated with tick bites and sorting out some of the basic mechanisms of that. And this is a real issue, in fact, in the area around where Cheryl works on the North Shore in particular. I'm sure you'll find her talk fascinating. So please welcome to the lectern, Professor Cheryl Van Noonen.
So today what I'd like to do is tell you a little bit about ticks. And a lot of you may already know what I tell you, but if you don't, then it'll help you understand the tick a little bit. And then I'd like to tell you about what can happen if you're bitten by a tick from the point of view of developing allergies. Then I'd like to keep you all safe by showing you how to prevent and manage tick bites. And after that, I'd like to tell you why it's such a fascination for people, for immunologists around the world, and for allergic diseases physicians. It's just allergy in a kit. How to get it and how to lose it. And there's nothing else like it allergically on the planet. So I hope you enjoy it this morning. Now, ticks in Australia, throughout the world, there are about 800 different species. And in Australia, we have around 70 species of tick. And 16 of them are out for our blood, literally. 95% of the bites that humans are given, however, from ticks are due to a single species called Ixodes holocyclus. And Ixodes holocyclus is known as the Australian paralysis tick. That's its official common name. Now, it's a busy little tick. It likes to inhabit everywhere along the east coast, from above Cooktown, as you can see, across to Normanton, right down the coast, and down to Lake's Entrance in Victoria. Sorry. It's still difficult to hear, so I'm just going to turn you up a tiny bit. Okay, I'll let you do it. Is that better?
Off the back? Right.
Yes? So this tick you can find anywhere in this area that's shaded in green. So you can see if you wander west of the Divide, you can have problems around Armidale, you can have problems around Canberra. And if you cross to our Tasmanian island, then you can have problems around Launceston as well, because this tick is in those spots. And all down the East Coast, the reason that it hugs the East Coast is that it loves humidity. It's there because it's nice and moist. And if we dry a tick out, it perishes. And their mothers put little drops of sunscreen on the babies so that they survive. All 2,000 to 3,000 of them, so it's rather a big effort, I think. It makes me feel totally slack in regard to what I've done for my children. So this is a picture of an engorged tick. So I don't like to really say this to the ladies in the audience, but she is the problem, because we're only bitten by female ticks. The male tick, however, it'll make many of the men uncomfortable in the audience to know that the male tick gets its blood feed by just drilling into the female's shell. So it's quite a story already, isn't it? This is the life cycle, as you can see. So it starts out with the 2,000 to 3,000 eggs, and then it becomes the larval stage, which has got 6 little legs, and then another blood feed later, and it's a nymph, and the nymphs are commonly called grass ticks, as most of you all know. Then it goes on to the adult, usually in spring, but any one of these types of ticks can be found at any time throughout the year. So I've had adults bitten by adult ticks when they should have been bitten by a larval tick, and vice versa. Now you can see over on the right, the female's on the left of those 2 large ticks on the top, and she has that hypostome or feeding tube out the front and the palps to sort of make it settle on the skin, just like you see with a crane and they jack it up a bit. The palps hold the hypostome in place. On the other side, you can see the male on the right doesn't need as long a hypostome because he doesn't have to go through human skin. He just has to find his woman. And he's right, you see, like most men in life. So now we'll move on to allergic conditions caused by the ticks, and vice versa, I might add. So allergic conditions caused by the ticks. The least problematic from the point of view of your immunologist and allergist worrying about you is to have a large local reaction. The most troublesome for a lot of people is that large local reaction. So people can have a tick bite and it can swell, and that swelling can go from here down to here, half their torso. Normally they're a bit more restrained, and it's about 5 by 5 centimetres, that sort of size of woody, red, itchy, burning skin, which often people think might be infective, so you generally score some antibiotics to try and take care of that as well. Now, more worrying for us is the fact that the tick is a very peculiar creature in that it is so good at causing allergy in people. It literally changes your immune system to do that. It renders your immune system prone to allergy. In other words, it can flick you into being what we call a Th2 or a T cell helper cell type 2 predominant person. Even if you didn't mean to be, you can be made such. And there's an animal model of this. Now, what can happen to, well, hundreds of people that I've seen, I've only seen 4 this week, in 2 days of seeing people. The tick bites you, it injects into you, and people make an allergic response to the proteins, one of the proteins in the tick saliva. And the next time they're bitten, when they disturb that tick, scratch it out, pull it out, use something from the vets, otherwise disturb the tick, they have an anaphylaxis. And these are profound anaphylaxis. The thing about tick anaphylaxis is it preferentially likes mature people. Most people are over 50. And it gives people a very profound anaphylaxis. We rate it from a grade 1 through to a grade 4, with grade 4 being, well, we hope everything's up to date in your will, because you might not make it. And these people have grade 3 or grade 4 reactions quite typically, so it's very worrying. Totally preventable, I'm pleased to say, though. Now, mammalian meat anaphylaxis. This is the entity that has grasped the imagination of allergic diseases people around the world. So the strange thing about this is that normally, as you know, we all tolerate mammalian meat. We may choose not to eat it, but we all tolerate it. And that's because it's a good source of protein. It'd be a shame if we didn't, because we build bodies with protein. Great source of iron, etc. But I won't take over where the Meat and Livestock Corporation could take you and give an advertisement for mammalian meat. But we're tolerant of it. But we lose our tolerance to that after a tick bite. Not everybody. This happens in a group of people who are prone to this occurring, but the mechanism will be that the tick provokes or promotes allergy proneness, and then we lose our tolerance to the meat, and then that follows on tick bites. It's usually a month or more after, it can be years after. And what happens then is that when somebody has their steak or their pork chop or whatever at about 7 o'clock at night, they wake up in the middle of the night having what I call a drop-down, drag-em-out anaphylaxis. In other words, you have to call 000, they collapse in the bathroom, and it's really quite a severe anaphylaxis again, often. So these two things are quite dangerous. I've seen over 600 people with mammalian meat anaphylaxis over the last 10 years or so. I see on average 1 or 2 people each week. Again, what I think is a preventable allergy, so that's our thrust, to prevent it. The large local reactions, I know we're missing an arm here. Normally we have a big fat arm, it might come up in a while, maybe if I press again, the arm will appear. I'm an experiential learner, as you've guessed. So you can see that huge reaction there to the tick bite, and you can see 2 tick bites. That's the sort of thing a large local reaction is, quite enormous really. It's all inflammation, it's all due to allergy, and it's all making a big mess of the arm. So as I said, you often get antibiotics for it, and usually a tetanus shot as well. So least problematic from the point of view of survival of the sufferer, but quite a problem. They don't start until 4 to 12 hours after the bite, because you have to gather your immunological brigade to do the inflammation, draw them in through the blood vessels, line them up, orchestrate the reaction, and then go for it with the inflammation. And then they take at least 10 days to resolve, and typically no after effects, as you can see. You look back on that arm in 2 weeks' time, you wouldn't know that that had been there. They occur with lots of insects, bees, wasps, mosquitoes, midges, you name it. You can have march flies, you can have a large local reaction to them. And probably that relates to the fact that those creatures are injecting a little something into us. Now, the tick-induced anaphylaxis is no different to, apart from the fact that it's usually more severe, it's no different to anaphylaxis which is severe from any other cause, peanut or prawn or blueberry or apple or pear or any of the things that I've seen, tomato, strawberry, you name it, I'm sure we've seen it. And if I haven't, as I say to my patients, that's why I keep turning up, because there's something new every week. So generalized itch is often the first thing. A feeling of warmth goes through the body. People feel as though they've been injected. And the important thing about this is it doesn't happen until you disturb the tick. If you leave that tick there, it could drop off having had its blood feed, and you would have no problem with it. So it's when it injects you because it's been disturbed. So it's a very nice thing, a tick, if you think about it. When it hops on, it races up about 4 hours it takes it to reach your hairline, it's what it's aiming for, and then it gives you a little bit of local anesthetic, and digs in with the hyperstome, and that sounds very kindly of the tick, but in actual fact the tick doesn't want you to know it's there, so it's purely a survival mechanism to give you the local anaesthetic. Then it puts its anticoagulant in because it wants its blood lake, because these are what we call telophagous feeders or blood lake feeders. Not like the mosquito. Mosquito just goes in, zaps the tiniest of little veins called a venule and then whizzes off. But this lady needs a lot of protein to make her 3,000 babies, so she needs a big lake of blood. The urticaria or welts may come up all over the person. Breathing difficulty, the throat can close. A lot of people have a feeling of impending doom, basically because there is impending doom. And nature has wired us to pick up on this in case there's something we can run away from to fix the problem. Dizziness, which portends collapse. In other words, your cardiovascular system just goes whoosh and every bit of blood goes into what we call the periphery or your skin and things like that. So you lose your blood pressure to the head, and again nature organises it so that at that point you fall over so you get some blood to the head when you lie down and you can think your way out of the problem. That's my sort of anthropomorphic sense of science. It's probably outraging Rob and Stuart down here, but we'll find out about that later. And some people even lose consciousness. So generally, I think it's important to appreciate this. You see, they're rarely fatal. There have been 2 fatalities of which I'm aware, none of them my patients, I'm pleased to say. 2 men in Queensland, and they awakened at night and disturbed a tick and then had a fatal anaphylaxis. Now, the life-threatening reactions are much more common than we see to anything else. From 2011 to 2013, I would see 1 paper wasp sting, 1 European wasp sting that had caused anaphylaxis, 2 bee stings, but 49 people with tick anaphylaxis. They would be the ratios. So that's extraordinary when you consider that bee venom is considered to be reasonably common as a venom allergy, for example. Obviously, it's more common than a shark attack or being struck by lightning as well. So that doesn't mean we'll go out and put our umbrella up, of course. But with tick anaphylaxis, this is what I was talking to you about earlier. I looked at 78 of our people and found that more than half are over 50. More than almost 20% are over 70, and that may relate to how many people have time for gardening. But I don't think so, because we get a lot of other people who are gardening, or a lot of people who are walking through their leaf litter backyard to put the clothing out or to bring the clothing in. So I think it actually is a situation where mature people are particularly attractive to them. And the male to female ratio is pretty close, but a slight preponderance for the men. And then again, that might mean that the men are out there doing a bit more of the sort of shifting the leaf litter, etc. Now, this association between tick bites and mammalian meat allergy was first described in 2007 because it was getting dangerous. I was seeing too many people. Like most clinicians, we have to be prodded into writing things up. And so I thought, this is a bit dangerous. There are probably people all along the East Coast who need to know about this. And so I wrote it up as an abstract, which was published online in November this month, actually 7 years ago. Now, we're very lucky with this because the Americans picked up on it. And Tom Platts-Mills is a fellow who's researched allergy for all his life, and he's been made a fellow of the Royal Society for his work on dust mite. In actual fact, he took an interest in this from a different aspect. They had people who were having terrible allergic reactions, again, grade 3, grade 4 anaphylaxis to an oncology agent for colon and head and neck cancer. And they just had 10% of people would have a really nasty reaction. And we've had a death here in Australia from it, in these people who are mammalian meat positive, because it's actually cross-reacting. The alpha-gal, or the little part of the molecule that people are allergic to, is in that oncology drug, and it's administered a whack of it intravenously, and that's an excellent way, unfortunately, to provoke a life-threatening reaction. So we're very lucky to know that the actual little bit of the meat that people react to is two sugar molecules or galactose molecules stuck together by an enzyme that the human no longer has. The great ape no longer has it, and the old world monkey no longer has it. So technically, these are the only meat our allergic people, our mammalian meat people who are allergic to the alpha-gal can eat. So it's either illegal, immoral, or hard to get. So it's rare that carbohydrates cause anaphylaxis. We get them in plants and things like that, but usually the allergens that cause anaphylaxis are protein in nature. So this is a curious allergen and a curious cause of the allergy, and a very common problem where I practice. So mammalian meat allergy, if you said to me, how many people do you see with mammalian meat allergy that haven't been bitten by a tick? I can number it on the fingers of no fingers, okay? None. Sometimes you see a little child who's meat allergic because they're intensely cow's milk allergic. And that occurs, and I've seen 2 or 3 of those, but that's it. So it doesn't happen unless the tick has bitten someone for practical purposes. The tick bite, of course, comes before the mammalian meat allergy because it has to condition the person to be allergic. And often they've had a little reaction to the tick bite in the past. Whoops. Now, often it's several months before it starts. It's typically delayed. So this is one of the difficulties, you see, because when you've got a protein allergen that's causing an anaphylaxis, it's always 0 to 30 minutes. Prawn can be 2 hours, egg can be 2 hours, but that's quite unusual even in those allergens. So this is quite odd. And the reason for that, it has to be metabolized and then come up through the lymph glands and the lymph vessels and then get dumped back into your veins at the level of the heart and go round again. And so this is why it takes quite a while. Of course, the amplifying factors for all allergies still are in force. I always say to people, if you really want to know how allergic you are to your prawn or your peanut, then what you do is you stuff yourself with it, you have it with chili, you have it heavily cooked if it's peanut and lightly cooked if it's any other allergen. You walk to the restaurant or run around the block, you have a headache tablet the day before, and you be slightly unwell if you possibly can at the same time, and then you'll know how big a reaction you can have, because all of those factors are tipping points for having an anaphylaxis or not having an anaphylaxis. And that's one of the reasons that allergy has this flavour of being difficult to understand. It's not. It just follows these rules of certain things making it more likely that you'll react or not react. So now this last line here is very important. Around the world, the commonest reason for an adult carrying an EpiPen for anaphylaxis to a food is peanut allergy. And where the tick is, and the people who live in what I call hyperendemic areas like the northern beaches, then it exceeds that. It's actually 0.12%, so it's more common than the commonest allergy needing an EpiPen. Now, the prevention of tick-induced allergies is really the prevention and management of tick bites. These allergens, to my view, are preventable. I look forward to the day when the efforts of Tiara, the Tick-Induced Allergies Research and Awareness outfit and everything that we're doing means that I sit back and wonder why I haven't seen somebody with that for a month. I'm looking forward to it, as I said. So now we'll go into the importance of the prevention of tick bites. So tick anaphylaxis is very rare outside Australia. There's one case in North America, there are a few people in Spain, there are a couple of people around the world, but it's really rare. And it might relate to the fact that our tick's feeding tube is the longest of all ticks, so it might get a better go at injecting what it has to change us. In Australia, we've got very likely the highest prevalence of the mammalian meat allergy, In the southeastern United States, it's very common, and they estimate that they have 5,000 people, which is more people than we have, but their rate is 1 in 8,000, whereas our rate here is about 1 in 880 when I think about the number of people who live where I live and the number of people I and my colleagues have seen with the problem. So there's some evidence from the US with their different tick, Amblyomma americanum, that if they don't get tick bites, then you might lose your mammalian meat allergy, because the immune system is always trying to fix it. It's always trying. So, for example, if you're penicillin allergic, 1 month after you have your reaction, you'll be positive on the skin or the blood or both. 10 years later, only 10% of people will be. Those 10%, unfortunately, might go off like a bomb, so we generally tell you to keep avoiding it. So it's trying to get back to neutral, and if we don't have a tick bite, then we might get back to it in 90% of instances would be reasonable. And there is certainly, I spoke to a woman last week who that had happened to. She'd had tick bites here, developed mammalian meat allergy, went to Europe, didn't get tick bites in Europe, lost the mammalian meat allergy, doesn't live in a tick-prone area and remains free of her mammalian meat allergy. Obviously, she needs to move back to the northern beaches to be scientific about it and see if she can get it again and lose it again, but she's been loath to do so for some reason. The other part is the importance of the appropriate management of tick bites and the treatment of tick-induced allergies. So with the anaphylaxis, we had a look at 78 people, 65 of them could tell us a complete story. And what we saw was, as I said, the anaphylaxis is so much more likely to be more severe. 75% virtually have a really nasty anaphylaxis. Everybody who killed the tick where it was didn't have an anaphylaxis after we made this diagnosis. Tick removal technique was known in 65. Everybody either pulled it out, used the household tweezers, scratched it out, disturbed it while they're awake, disturbed it while they're asleep, disturbed it in the shower because they didn't realise they had a tick on their back, or used something from the vet. So potentially avoidable in really the vast majority of instances, I believe. So I'll go on from that. So the Australasian Society, these are our 2014 recommendations, and they will be our 2015 recommendations as well. Kill the tick where it is. Use an ether-containing spray to freeze it, because it's dead immediately then. Wait for the tick to drop off, or remove it without compressing it. You see, you can still kill the tick, and it's like a bee. If you've got a dead bee, you can still sting yourself, can't you, by squashing your foot onto that bee? Almost everybody in the room's probably done that. Do it in a safe setting. If you've had an allergic reaction before, take yourself up to the emergency department, but do it yourself when you get there so you know you can do it. And use a tickercide, which is used for scabies generally, called Lyclear or permethrin cream, to kill the little ones that you see, because it's really hard to sort of zap them accurately, and you can't sort of zap something that freezes your skin 40 times because you might burn your skin with the ether spray. So it comes down to 3 easy things to remember. For a larval or nymph stage tick, dab it, don't grab it. Use the permethrin cream. For an adult tick, freeze it, don't squeeze it. Use the ether spray. And then the other thing to remember is household tweezers are tick squeezers. So they will squeeze the allergen into you, and that is not a good idea, as we've heard this morning. Now, common advice about tick removal has been fine-tip tweezers. Now, they will work provided you get right under and you take it up, but a lot of people have told me, remember the demographic that the tick anaphylaxis favours, and a lot of my patients said to me, my husband or my wife can't see to use the fine-tip forceps. They're hard to get, and I think that really they're not useful to us in the general run of tick removal. Fine in the emergency department. And that's, they say, grasp the tick, remove it immediately. No, leave the tick in. If you're by yourself, leave the tick in until there's somebody home in case you have an allergic reaction to it. If they're home within 16 hours, you won't get a nasty illness from it either, because it doesn't start until the tick's been in for about 16 hours. Rapid removal reduces the risk of infectious disease, yes, but you don't want to get an allergy from it either, so we have to do it when we can, within the time, as I said, 16 hours. Disadvantages to the usual advice is that they're not widely available, they're substituted with household tweezers, which makes the whole thing dangerous, rarely done properly, difficult to achieve in young children, and as I said, eyesight can be a problem. Not useful for removing the nymphs either, usually the little ones, the 2 millimetres. So now I'd like to tell you about the significance of the tick-induced allergies, because it's really fascinating that this allergy or allergies, they represent, if you could design a kit to tell you how to make an allergy and lose it too, then this is perfect. So it's very insightful into what happens in our immune systems. Firstly, the tick bite, as I said, promotes allergy proneness in the host. There's an animal model for that. Maybe it's because the tick's there for a while that helps, so it's got time to modulate the immune system, unlike the mosquito. The severity of alpha-gal allergy, or that meat allergy, is also of great interest to us. These things are both very severe allergies in the majority of people, so this again comes back to the idea that the tick is altering us. And 2 severe allergies from the one provocation, protein and carbohydrate. And that's probably because the protein presents the carbohydrate, the sugar, the alpha-gal to the immune system. Ticks inject the allergen. If you want to become allergic to something, injection's best. Eating it will partly tolerize you because we have this wonderful arrangement in our mouths of dendritic cells which will start making you tolerate that substance within 15 minutes. Ticks provide the allergen exposure. Not only do they make you allergic, injected, they give you the allergen and tell you which allergen you should direct your efforts against. And then we're left with the conclusion, aren't we, that if we avoid the promoting factor, and the allergy may remit because that's what allergies do, then there's a role for secondary prevention. In other words, if you've got the problem, maybe not having a tick bite will allow it to go away. But then we've got primary prevention possibility as well, in that if you deal with the tick correctly, then you don't get sensitized to the tick salivary protein and have a tick anaphylaxis, and you don't get sensitized or lose your tolerance for the mammalian meat allergen and don't become allergic to mammalian meat. So that's why I sort of term it the How to Make an Allergy and Lose It kit. Now, we've set up Tiara as well, which is aimed at promoting awareness, and that's been a wonderful medium with a website to try and get this information out, because when we saw on that first slide, we see how extensive the area is, and those areas do not have an allergic diseases physician on their doorstep. So we've put up the diets that people need, the iron intake advice that they need to make their lives comfortable if they're living in Cairns, as one of my patients does, or Tambourine Mountain. As long as they can get onto the internet, then they can keep themselves safe. And so thank you for your attention today, and I hope you've enjoyed the morning.
Thank you very much, Cheryl. That's fantastic work that Cheryl does. And if you'd like one of the pamphlets, there are some down the front on the way out, and I think there'll be some on the way out the front door. I'm sure you'll be all going off to have your green salad and get some ether spray afterwards. Okay, it's my great pleasure to introduce our last speaker for today. This is one of our Garvan superstars, I think it's fair to say. Professor Stuart Tangi works in the immunology department. He works in the area of human immunology, particularly in the realms of immunodeficiency, and it's really won numerous awards over the years, including he's a principal research fellow at the National Health and Medical Research Council, and he won one of the great prizes in Australian science, which is the Gottschalk Medal from the Australian Academy of Science, and we're very glad to have him in our program. And he's going to talk to us today about his work on how studies of rare diseases impact on medical advances for the masses. Please welcome Stuart Taylor.
Thanks very much, Rob, and thanks to everyone for coming to Garvan today to hear about some of the work that's going on and to give you a bit of education about immunology. Just get my... Yep. Okay. Okay. So as Rob said, I work on immunodeficiencies, and we'll get to that in a sec. But what I just want to give you an introduction to is obviously the immune system. Yes. In general, like the immune system, like every system in our body, when it works, you don't appreciate how well it works. So obviously what the immune system does, it's really the fortress of our castle, which is our body, of course, to keep us healthy by preventing disease, particularly infectious disease. So the immune system does some wonderful things. It's like, as I said, it's pretty much an army of helper cells which can do things like have control and initiate inflammatory responses. We have other cells which I work on, as does Rob, the B cells which make antibodies, and the antibodies are the proteins which protect us against infection. We have other cells of the immune system which go in and kill cells that have been infected with pathogens, particularly viruses. And there's other fantastic cells of the immune system which gobble up a whole lot of infected cells also. So these are the guys that we're trying to protect us from, these different sort of microbes, bacteria, viruses, fungi, mycobacteria, all these sorts of baddies that are out there, whose real mission is to survive themselves, but of course to do that they need to infect hosts. So I think looking at this slide here, what this shows us is actually the lifespan over thousands of years, expected lifespan over thousands of years. So just ignore the red line for a second, but all these other lines pretty much overlap. And that's drawn from historical data going actually back to thousands of years pre-BC, which predicted that the lifespan then, up until about the 1800s, was only about 25 years of age or so for 40 or 50% of people. So myself, and probably everyone in this audience, if we were alive 200 years ago, we probably wouldn't be alive at all, because the life expectancy was so short. And that was really due to the prevalence of infectious disease and essentially our inability to control these different types of infections. But what you know about today, of course, at least in the UK 15 years ago, our life expectancy today is 80 years or plus or so. So what's happened between the Stone Age and today? And it's not as if the body's suddenly acquired a whole lot of super immune genes which keep us alive. What's really changed is three fundamental changes to life which impact on the immune system. The first one was the introduction of improvements in hygiene. This was in the 1800s, the simple fact of washing your hands, keeping yourself clean. That was the first, which today, of course, it's hard to believe we didn't do that. But the awareness of hygiene was important. The next one, of course, was the development and implementation of incredibly successful vaccination programs, and then the introduction in the 1900s of antibiotics. So the collections of these three advances led to minimizing infection by improved hygiene, treating infection with the development of antibiotics, and preventing infection by the development and implementation of vaccines. But as you've already heard from the talks this morning and just now, the immune system can also go a little bit off the rails. And that's really the focus of a lot of the work that's done here and, of course, many other places. So while the immune system is fantastic at keeping us healthy, sometimes things go not quite the way they're supposed to. And that can result in, as Cheryl's just told us, and Rob spoke earlier about things like asthma and allergy, which we hear a lot about. There's many autoimmune diseases, systemic lupus erythematosus, inflammatory bowel disease, diabetes, arthritis, and many, many others, of course, multiple sclerosis. There's types of cancer, so the immune system not only protects us against infection, but because some types of infection can cause cancer, the immune system is actually very important for what's called tumor surveillance, which keeps a lookout for cells that have become cancerous. But also we can have cancer of immune cells themselves. So things like lymphoma, which we hear a lot about, leukemia, these are all cancers of the blood cells. And then what I want to talk about briefly, or highlight some of the work that I do, is in this field of immunodeficiency. And we hear a lot about immunodeficiency in the context of HIV infection, for instance. That's an acquired immune deficiency, which, as the name suggests, you acquire it due to an infection. There's also other types of acquired immune deficiencies that can occur when you, for instance, are stressed, or when you get poisoned or have chemotherapy or things like this. But what I work on are actually genetic diseases. So these are diseases which are caused by mutations in single genes. So these are called primary immunodeficiencies, sorry about that, primary immunodeficiencies, and they're often referred to as experiments of nature. These are naturally occurring conditions which happen because there's a mistake or an error in a single gene. So in our genome, there's about 25 or 30,000 genes. And a lot of those genes we can actually function quite happily without, because there's lots of ways of making up for those mistakes. But there's about 250 genes have been identified, and actually I was at a meeting a few weeks ago where they report on the latest findings of genes. So this number will increases almost every week, to be honest. But there's been about 250 genes identified out of our genome of 25,000, so about at least 1% of our genes are absolutely critical for our immune system to operate, because if you lack one of these genes, you can have what's called a primary immunodeficiency. Now, the good thing is these conditions are incredibly rare. They can affect about 1 in 100,000 to literally 1 or 2 individuals in the world. So they're incredibly rare conditions, which is obviously a good thing. However, they're incredibly informative as well, because we know these patients get very sick. They present to clinicians with recurring infections, usually from a very young age. So obviously they appear early on childhood. But they're incredibly informative and educational to us, because what they do, they can tell us why those kids get sick so dramatically. They can inform us about how we can develop new treatments for these diseases. It tells us what those genes do and why they're so important and why they can't be replaced by other genes. And it can lead to the development of new ways of treating these conditions as well. So to highlight this, I want to give you an example of 2 boys. One you may recognize. This is me as a 7-year-old. As you can see, I'm a fairly healthy, strapping young lad. And by that stage of my life, I was in school, I was playing sport, I was probably rolling around in the dirt, I was swimming, I was getting hugs from my mother. But this little guy was born not long after me, actually. His name's David Vetter. And you probably better know him as the boy in the bubble. He was a real person. He was born in 1970, just after I was. But unlike myself, this is David, when he was about 7, he spent all of his 12 and a half years literally confined in a sterile plastic bubble. Now, there's obviously ethics and moral questions about whether that was right or not. But he didn't have any of those childhood experiences that I did. He didn't actually get held by his mother until he was 7 years of age. And then he tragically died when he was about 12 and a half, following a failed bone marrow transplant. But the main difference between myself and David, obviously apart from the geographical ones and so on, was one gene. He had a gene, an error in a gene, and that caused a disease called X-linked severe combined immunodeficiency. So he had one gene that didn't work, and that rendered him susceptible to almost every pathogen that you could name: bacteria, viruses, fungi, mycobacteria, lots of things that we deal with on a day-to-day basis and don't even realize we're infected with, like our skin, for instance, believe it or not, is crawling with bacteria. All these sorts of things really compromise the immune system, and that's why David spent his life in a bubble. So I work on a disease, number of diseases, but one is called X-linked lymphoproliferative disease. Now the name doesn't really have to resonate with you at all. One point to make though is it's X-linked, and Cheryl made a comment earlier, which we may echo here. It's a disease like the disease that David Vedder had. It's carried by the mums. So the mothers are healthy, but they pass it on only to their sons. So 50% of boys born to carrier mums can have this disease or have any X-linked disease, like colour blindness and so on. So it's carried by the mums, passed on to the boys. And what's unique about this disease is that it's triggered really by exposure to a pathogen or a virus called Epstein-Barr virus. Now, you may have heard of that, but if you haven't, you've probably got Epstein-Barr virus. This is a virus which is ubiquitous. It's in more than 90% of our population in the Western world. And most of us get infected, and most of us deal with the infection without even knowing we're infected. And if you do get sick, the worst thing that can happen is you can develop glandular fever, which is also called the kissing disease, because that's how it's transmitted, usually by those active immunosuppressed high school and university students. So what we do, we work with boys and their families with this disease. We study their immune cells. We look at proteins in their cells because they don't have this particular protein here. We can see it's missing. We look in their DNA and work out why their protein can't be expressed, and we do lots of studies to try to understand why the lack of this single protein really compromises these kids' health to a virus, which is really quite common. Now, as my title suggested, we study diseases in rare groups, but we try to have our studies have great applicability to the masses. And that's shown here. So I'm always asked, oh, well, you study these rare diseases, how can that possibly help me? And that's not an unreasonable question. But Epstein-Barr virus, as I said, it causes glandular fever. Now, that's not so bad. I had glandular fever when I was 21, and I obviously survived. But it's a bit of a nuisance. But that's not all. EBV was discovered 50 years ago, 1964, to be the first pathogen that can cause cancer. So it's what's called oncogenic or cancer-causing. And EBV is strongly associated with many of these malignancies listed here: Burkitt's lymphoma, Hodgkin's lymphoma, non-Hodgkin's lymphoma, Bcell lymphoma, and a few other more fancy names like nasopharyngeal carcinoma, which is actually quite common in Asians, and leiomyosarcoma, which is a cancer of muscle cells. So it's incredibly problematic in some different demographics and some different ethnic backgrounds. And obviously it's absolutely causative of this disease that I work on. It's actually questioned. It's implicated, but not really sure whether it's involved in these. But these two columns here really highlight the applicability of here. So what most studies are focused on is studying these rare patients with these problems with dealing with infections, which can actually be problematic in the greater society. So by studying these boys and their families, hopefully we'll be able to work out ways of boosting immunity to things like Epstein-Barr virus, but also developing a vaccine, because as I said, EBV causes glandular fever, infectious mononucleosis, but can also cause numbers of cancers. So if we can prevent infection with EBV, we can prevent cancer. And of course, there's a great precedent for that. And we all know about Gardasil, which is the vaccine developed by Ian Fraser and his colleagues in the US, which is the first vaccine against a cancer. And that works because it prevents infection with human papillomavirus, which is a virus that causes warts, amongst other things. But of course, it can cause cervical cancer. So we can actually vaccinate against cancer by targeting the immune system against a virus, which we know can cause malignancies. So, and I should say, there's no vaccine against EBV. So if we can use our knowledge to convert that to a vaccine, we can treat lots of conditions and maybe prevent some cancers. So there's lots of reasons to study the immune system, and you hear about this in the news all the time, whether you appreciate it or not. Most recently, we've heard a lot about Ebola virus, obviously, rapid, almost desperate measures to try to implement a vaccine for that. A few years ago, it was bird flu. There's always things like HIV, malaria, tuberculosis that are being discussed about how these infections don't have good vaccines against them. And then there's other diseases which affect different populations of people, as I mentioned, autoimmune diseases, cancer, asthma, and my favorite condition over here, of course. So it's really valuable to study the immune system because really without it, you're prone to all sorts of infections and other conditions. And with it, if it goes too wrong, you can have all these sorts of conditions happening as well. Thanks.
Thank you very much, Stu. Well, that brings us to the end of the seminar today. I hope you enjoyed all the talks we had today. Thank you very much to our speakers for presenting. Again, if you'd like to be in contact with the Foundation or any of us, please do get in touch. Otherwise, thank you very much for coming today and enjoy the rest of your day. Thank you.